So in the document, there's obviously the green bit at the top, which reflected what we've done in the first session. And then there's like these little outcome boxes. And I've duplicated one for us, which I just called subtopic 3. And just above it, I've pasted the points that were on the slide, just because I think it'll help to remind us what we're trying to see. But I just thought
is this
for you to know where this is.
Is this in the document 16D on the Google Doc?
Yes.
Okay, great. I just wanted to make sure I was looking at the right one.
Yeah, so if you actually the easiest way of getting there is scroll back up from the bottom is the first thing you'll hit.
Yes, right. Got it. I was just trying to figure out how to sort through all of this.
Yeah.
And then so for now, I'll just fill out things in the green section, and we'll try to make sure that we're answering the questions that they're asking up above, which is, what are the problems we think we can solve by collaborating? Is that?
Yes, so I think within each of those 4 points, we have to see whether we can find one particular point where we feel that by collaborating, that we can identify a question, a sort of specific sub-question where we can make a difference by collaborating in one way or the other, or where we can maybe identify a group of people to make contact with, or anything of that kind, or start a little sub-consortia, or whatever it may be.
Got it. So I'm just
great. I think it's 20 past, so I think we might as well make a start. So just in case people have just joined, in the initial outline, I basically had 4 general points that related to, hang on, where my
I've just modified the document. Is that okay?
Yeah, so there's the first point in the subtopic was if we think of what our ideal cell type annotation looks like, there are a number of things that we'd like to see. So the cell type annotations have been hierarchical. We'd like to see some reflection of cellular plasticity, some reflection of spatial localization. We'd like to take into account classical histology, and of course link with existing ontologies. And I think what would be really useful for us as a group to now kind of say, which of these do we actually think are like a bottleneck? Where do we feel that there's something that's holding us back in terms of getting effective cell type annotation? So please feel free to chip in.
Identifying ontologies that we can use cross species and cross modalities seems to be key.
Yeah, and I agree, but it means that it's the devil's in the details. It is pulling up those ontologies and going through them line by line. And this probably would be best to be done by, you know, a group of individuals that have been not vetted, but in which people are comfortable that these are the content experts to be able to go through those ontologies, as I said, line by line. It would probably be useful to do that as a collaboration between Human Cell Atlas, HubMap, and probably KPMP, GoodMap RBK. At least for kidney, we're already starting that. Probably, you know, getting lung map folks involved and any other organ-specific consortiums involved.
Yes, I completely agree. I think when it comes to ontologies, what really needs to happen is that groups of people send in their annotated data, and then a smaller group really looks through them, tries to combine them, but then come up with a sort of newly combined suggested annotation that is fed back out to all the different collaborators, and people have a chance to comment on that. And of course, meeting in person is really helpful for that, but that might be quite difficult in the near future. And I wondered, I saw that Martijn Nabijn is also in this meeting, and I just wondered, Martijn, we've obviously just started a big effort to do this in the lung, whether there are particular observations that you might have that we, you know, just from your experience over the last 2-3 weeks of what might work well in other organ systems?
Yeah, thanks, Kerstin. I think from the experience we've had of integrating all the data sets that we use to do the COVID-19 analyses that were presented yesterday, we were working with something like 25 data sets, all annotated by the lab who contributed the data set. So at different levels of granularity, using different semantics, if you wish, or different language, not agreeing usually on the basis of annotations, and definitely none of them was referencing back to existing ontologies in great detail. And I think that's going to be the situation for a while, at least in most of the organ systems within the HCA or other initiatives that try to do this similar things, where people will annotate the cells based on their previous knowledge, on their interest, and not so much based on highly systematic ontology. So in that sense, I think for any ontology to be useful, it should be really data driven and easy to use for people who do not have an ontology interest per se, but just view this as a tool to be able to integrate data sets across studies, even across organisms, which is yet another level of complexity, or even across developmental stages, if you wish. So in that sense, I think the ontology should really be practical and pragmatic, even sometimes, where and not so much trying to get every detail completely right and then having that set in stone, because that doesn't help anybody. I think the ontology, at least as I see it, should really be a tool to advance the field and to allow us to integrate data sets, because that's at the moment where a lot of the hurdles come from. If you start comparing data sets, we're talking a different language, and that needs to be integrated.
Yeah, I think those are great points. I'm sorry, David, I just wanted to make one quick point that those are great points. One of the problems with getting line by line through ontologies is that you do tend to get down into the weeds. And one of our interests at the Broad is to make sure we can do all that cross data set, exactly what you were just talking about. And identifying a minimal set of things that you can use across data sets seems to be a very useful concept.
Yeah. I think that also comes back to really making the most of the hierarchies within those ontologies, so that you know, things are definitely and I don't think people who manually annotate at the moment do that enough, that you first annotate that it's an immune cell and then a lymphocyte and then a T cell. You just put a T cell, and of course the ontologies should be able to sort that out for you. But if you don't use standard terms, that doesn't always happen, I don't think.
Yeah, I think that's a very good point, Kerstin. And I must say we had we were in the fortunate situation that we could actually use Mark Krasnow annotation of his lung data set. And he actually did this. He followed the hierarchical structure of an ontology. And I think he also referenced back to existing ontologies in his effort to do so. And that really helped. And that also was the backbone of the structure that we used to then integrate all the other ontologies onto, if you wish. So that was really helpful. So I think that's a great point that you make, Kerstin.
Yeah. Now, I've picked on Martijn because I knew that he's been working on the lung, but there are many other contributors here. Does anybody want to chip in from experience, maybe with their own data, their own organ system that they're looking at?
I want to have a follow-up question to Martijn. In your recent experience, I imagine it's easy to agree on the course labels, but it's a lot harder when you go too deeper. How do you guys decide when to stop, to say this is not important today?
That in our case, thanks for the question. It's an excellent point, because we were clearly struggling with this. At the end of the day, our question was about the ACE2 receptor and the TMPRSS2 protease gene expression across cell types, and then trying to analyze what's the effect of age or gender or smoking status on the gene expression levels. So in that sense, to be able to do this with some statistical power, we had to go back to not too fine-grained annotations. Because, so for instance, we did a lot with epithelial cells, and then in the lung, we basically said all the secretory cells we put into one bin. So it's, and all the ciliated cells we put into one bin. We can't start annotating the subtypes of ciliated epithelial cells, because that would be too fine-grained, and then the data sets would basically break apart. And then you'll have a contribution from one data set, but not the other. I think as we accumulate more data as a community, this will become a possibility. But there's also technical limitations with single cell data, in the sense that there's strong batch effects, center-to-center batch effects. So that all gets in the way of harmonizing annotations. So the way, to answer your question, the way we dealt with it is really to don't go down into too much detail. So for the epithelial cells, I think we ended up with 5 or 6 subsets of epithelial cells in the lung, and that's it. And that's where we stopped. And clearly there's more, but we just didn't address that.
Thank you.
Right, so maybe I'll just open the discussion back out to any other organs at this point.
Could I just make a quick comment that if anyone sees me missing anything in the notes, please feel free to contribute.
Great, yes, and I was scribing yesterday. I found that very helpful when others chipped in, so please do that. So as there's no volunteers for other organs, I mean, the session was about different modalities. So far, we've talked about the problems we already had when you know, annotating within 10x single cell data for the same organ. So that's as little cross modality as you can possibly get. Are there any people out here that maybe produce other types of data, proteomics, metabolomics, and you know, where they see the major problems in terms of integrating data?
Yeah, and for the BRAIN initiative, we have a lot of experience with this, but then also to an approach that's been taken both from the Allen Institute on its own, as well as in the context of the BRAIN initiative, and that's to generate intersecting modalities when possible. So the sort of Rosetta Stone approach to overlaying different modalities and then using those cross-modality data sets to link up, sort of defining truth amongst those modalities. So for example, if you have transcriptomic data, then for a limited set of exemplars, ideally that intersect with your nodes of cell types of interest, you collect cross-modality information.
So, and actually, I really like that point, which I think Kathy's been able to record, but because that seems to me like something where we can really make use of the consortium, so that if one particular group is maybe particularly good at one thing, but has spare tissue that they could make available to people using quite different modalities, then, you know, I think this forum really indicates that that would be a really valuable thing to do, so that we start to assemble these cross data sets. Of course, you know, in a way you would like to set out doing that in a very systematic way, but I think we just don't have the breadth to do that. So I think it has to initially come from sort of individual initiatives. So is there potentially, do people think that that is something that would move the field forward?
I think that a challenge to that, a very obvious challenge, is just as you said, there's methodological challenges of being able to access the tissue. And then there's sort of the challenge with the community of making others aware of what you have and what's available. So I think most people would recognize it's useful to have that kind of integration, but the challenges, if we were to identify an obstacle to getting to not just having everybody do that, it would be to understanding and sort of centralizing the information that is being generated anyway, and then where there's complementarity around other people's experiments that could use that information or build on it.
Yeah, so again, and from what you've just said, that actually is I think that is a problem that we can potentially solve by improving our collaborations. And I hate to come back to the lung, but within Europe, we've actually, the EU Horizon 2020 program very much compels you to work across multiple countries within Europe. And that has meant that we have actually put together a collaboration where we collect tissue at one source and then hope to profile that in a number of different modalities in different places, from sort of single cell to attack, and then a number of different spatial techniques, always sending the tissue back to those labs who are most expert in that particular technique. And hopefully that will actually be a good way forward.
So one of the, again, just practical impediments to that, Joshua Levin has this in the chat, but for some people that don't necessarily start with that ambition or haven't started with that ambition, some very practical concerns with MTAs and IRBs, like practical paperwork can be a huge issue. And even at the Allen Institute, when we work on brain, even within one institute, we sometimes have challenges with multi-purpose use of brain tissue data based on the source from which it is acquired from. So yeah. I completely agree. For lung and all these organic systems, people are encouraging and they want collaboration, but when it comes down to what is holding people back, it's not the will, it's the fact that there are practical impediments, and those include understanding who else is working on your data set and what state they're in, and then actually knowing and having active access to the tissue in question, which is sometimes prevented through very practical and mundane reasons that include tissue preparation protocol differences, the IRB, the permissions, the workflow associated with how you would even transport that material, sometimes particularly if it's international, all kinds of, like, again, as somebody had already said, the devil's in the details for this as well. And those are, you know, for focusing on what could improve, those are the impediments that I think there would need to be solutions identified or worked toward through a working group or an additional focus group to try to address some of these.
It also sounds to me as if people are willing to make that information available as part of the HCA. Obviously people put down what organ systems they're working on, but it sounds as if a list of more detailed projects might actually be very helpful to get people to make the right links to the people that work on a similar tissue, similar question, but with a different modality. So that is potentially something that, you know, where between HCA and HubMap, we could put something together. I wonder whether that could somehow actually already be identified as is sort of where we, you know, in our blue boxes, which say, you know, something that we can actually start to think about solving, whether we can record that in there. But I won't do that right now, but I completely agree. I think that was a very great contribution. Thank you.
Hi.
So to address Amy's concerns, KPMP has dealt with this whole issue of multiple sites needing a common IRB, common consents, and they're quite happy to share those documents.
Hello?
Okay, so a problem, I'm actually going to take us back to the little blue box because I think it is useful to record some of these ideas. So in the blue box, then we have one of the problems that we think we can solve by collaborating is actually identifying collaborators that have different techniques to apply So the same tissue, sorry, my typing here is terrible. The same
Don't worry.
tissue, we can fix that up later. And then as recommendations to make that possible, where would we find that information that you just volunteered? The kidney group, where do they have these?
You just need to contact Jonathan Himmelfarb, and he already made this effort, this offer in another subgroup. So we have KPP has shared this information with HubMap, and there's no problem of getting this these documents. So Jonathan Himmelfarb is the individual. And if there are any, if you need more clarification, you can contact me offline.
Great, okay.
Hi.
I'm sorry, I did write this in the green box. I didn't realize I should have put it in the blue
sorry.
box.
Yeah, excellent. Okay, so I'll stop recording anything. So can we then maybe—
Could I bring up something here? Can we bring up something real quick? When we are talking about different modalities, I have always had problem with single nuclei data not having the same ontology as single cells. Could we think of single nuclei as a new modality, please?
Can you just clarify a little bit? So in terms of your cell type annotations, you are getting quite different cell types when you're using single nuclear data.
Different gene lists to identify these cell types. So it would be unique, non-overlapping set for single nuclei data, which is not necessarily the same as single cell.
Right. Yeah.
We actually, in the lung efforts, we did have 1 or 2 single nuclear data sets as well. And even though the genes are not identical, calling out the cell types was doable. Of course, it takes somebody to know the cell types to be able to do this, but we were able to give them the same ontology annotations as the single cell data. Automated, of course, becomes an issue if you would want to do that, which is where we want to go, of course, because this just took me and a few other people a lot of time and a lot of effort to do it manually. So if you go automated, then single nuclear is going to be well, it needs some adaptation to the automated algorithm.
Sounds like that might be a useful second, offline discussion, but not necessarily one of the key things we want to include in this ontology discussion.
Yeah, I think that's.
So, Kerstin, I also identified a couple of problems that I thought I was capturing, that identifying ontologies that we can use cross-species, cross-modality was one of the problems that I think that we said we could solve. So I didn't put that in a blue box. I put it in the green box above just until we sort of zero in on that. So that's okay. All right.
Yeah, we'll leave it there for the moment, and then we can see whether we can maybe move it later.
So I would like to add to that, if you're going to look at the pre-existing ontologies, it seemed from the discussion that there was a real focus on developing an ontology which would be more appropriate for single cell, for having the molecular data. And so, yeah, I guess I'm hearing 2 things. Existing ontologies, again, you'd have to go line by line to see whether or not it's appropriate for all the molecular data. At the same time, do you want to use that as a partial framework to start with, or to develop a new ontology which is going to be molecularly based?
So if I could comment there, I think we have found it quite useful. So in the Krasno paper on the lung, they made a very careful effort to say, these are cell types that were described before, and which ones are we finding, and where are there cell types that we see in addition to that. And I think each organ now needs to work on expanding their own ontology, but I think it would be crazy to, forget about all the knowledge that has been accumulated over the last 20, 30 or even more years by different fields, because
Right.
there must be a very rich data source that, we don't want to
Yeah, no, and I agree. I mean, our experience with kidney is that as we start to go in and look at just the kidney anatomy aspects, that there's stuff in Uberon, which it's not clear why it's there. It doesn't make sense. If you look at the cell ontology, focusing again on kidney, there are a lot of things there which are just not fitting any longer. I mean, it may be because we've got new data or we have a better understanding. And that's why I made the comment about, using the current ontologies. It really means that you need to go line by line, or at least the portion of the ontologies you're going to input then to build onto needs to be looked at really carefully, because there's things there that just, as the ontologists have told me, anytime you only have one entry after you have one topic and then one entry under it, that there's something wrong there. So again, I mean, the word of caution is each organ really needs to have someone who will go through those ontologies carefully to figure out what is appropriate for using them for a molecular, building a molecular ontology on top of it.
And I guess from what you said, it sort of implied that this really has to be happening within organ biological networks, because obviously you want to have that as spread across your community as you can. So I guess we could just maybe record that we, strongly encourage these organ-specific biological networks. They don't necessarily help with some other sort of cross-organ annotation, but I think that would be a very good start. If I just look down the list of things we discussed earlier, do people have any thoughts of how spatial information could be included in ontologies?
Anoop had a comment that was in the chat about this that I thought was very helpful, or would be a good thing to focus on maybe. It was a little ways up. Can he weigh, is he still on? Can he weigh in on that?
Yeah, I was on mute, sorry about that. So are you referring to my comment about what we're doing in the hub map, or was it about the T-cell differentiation states?
Yeah.
The latter? Or,
no, that sounds great.
Okay. So we've been looking at the immune cell, different types of immune cells, and using the existing ontology. But when we look at the actual marker expression, we do find out additional things. And so how far do we go in defining these different immune cell types? So you have Tregs, but those only refer to the CD4 positive cells. But FOXP3 expression is also in CD8 positive cells. So there is no existing cell label for that.
Yes, and I guess we as a community have to start making those labels, and that, I guess the question for us as a group becomes, how can we do that in a concerted effort? What framework do we need to make that happen reasonably efficiently?
And then you have these different cell differentiation states, like the memory T cells, the effector T cells, all these different cell types that are very relevant for the cellular therapy space.
Yeah.
I'd like to add a comment, like a follow-up to what Deborah mentioned. I think, to paraphrase what she said, it is the need to map the new molecular taxonomy to the older, many multi-layered taxonomy. Some are based on histology, others morphology. So I think there is a potential danger that is when we felt we've mapped them well, we start to use the same names for those cell types, that others may not realize this name now means something new. So there might be years of confusion that somebody is using this in the old sense, others use it in the updated sense, and others use it in kind of as one word containing 10 layers of meaning, including spatial. Suddenly we have to kind of put in additional version to re-annotate the names.
Yes, and I agree completely. And I suppose in the early days of gene mapping, everybody gave their name gene their own favorite name. And then at the end of the day, the problem had to be solved by big community-led efforts to synchronize the naming conventions. And I guess somewhere down the line, we will be facing that as a field. I don't know whether we're still a little bit early for that. I don't know whether people feel we're ready to start something.
I hope we don't go down that same path.
So
what did you perceive as
control vocabulary is going to be absolutely essential. I mean, it's essential in terms of any search engine that you want, and to have an agreed upon nomenclature across consortia. I mean, I was amazed that even within the different projects within a given consortium, they're using different terminology for the same thing. And again, I see this as, what is it, crock up? This is one thing across the different consortia. You could break it down to individual organs, but the different consortia, working on a specific organ, but they need to all get together and agree upon the nomenclature for these various cell types. I mean, is it going to be sufficient to say that this cell is a KIT positive, something negative, something positive? We need a discussion to figure out some sort of process for naming these cells that everyone can agree upon.
I think it's the same in a sense that we at this point have the broken up discussion, because I think the automated cell type annotators might have a good way of judging the hierarchies at which you can agree on common names, and have a very unbiased way of, data-driven way of then generating names that may not be confusing with prior annotations. But alas, they have their own discussion, so we won't go down that route at the moment. But so do the best way forward, presumably, is once again meetings where people come together and actually try to share data and come up with naming conventions. Do people want to comment? Rayasri, did you want to comment?
Sorry, go ahead. I wanted to point out that I'm not sure a meeting is going to be, I mean, meetings are a part of it, but, looking back into a comment that Kathy had as well, I think that there's something that's a little more concrete that requires things like dedicated funding and committed people to work on an output that is usable and accessible to people across all of the different organ systems. So there's subspecialization within those organ systems, but people aren't using the incorrect terms or not having terms because they don't want them. It's that they're not available. There's not some sort of centralized source. So, go again, if we're thinking about the crocs up and what's a next step, it could be to seek or solicit or identify the need for a centralized ontological solution that is comprised of a funding mechanism, possibly a grant that goes together to articulate that overarching requirement that would have a number of people, staff, whose dedicated job it was to create and manage that, and then pull from the different organ groups as necessary. So I'm aware that a lot of the different sub-projects that have organ-specific attention have done that on their own. But as Kathy points out, there's something that's needed at a level above that. I think an umbrella above that, to which those organ systems can reconcile and iterate. the idea that there would be one sort of summit where everybody agrees, my experience is that the agreement is unlikely to happen, and it would quickly sort of time out for becoming obsolete as more data becomes available, as richer information is generated.
Do we have any representatives of funders in this Zoom room, or maybe representatives from repositories like the EBI or something like that who might like to comment in terms of how this could be helped? No, not by the looks of it. Okay.
No, but I think it would be a good thing if we just put it in the notes as a next step, you know, for reporting out as a next summary. Was it the blue box? I can't see it right now, but.
Yeah, so I've actually just done that. So
oh, good, okay.
So Amy, I think, you know, the fact that HubMap is being funded out of the Common Fund, the NIH Common Fund, and I think, you know, this is something which would not be necessarily supported by individual institutes or centers. NIDDK, my institute, may very well think, you know, maybe we want to do this just for kidney, but there's really needs to be something which is more of a institute-wide effort. And I think Richard is the one that we should be kind of tapping on his shoulder. And you can tell him I said that.
I think it goes above Richard is where I'm going. I'm going back again to this group of HCA and HubMap and beyond.
Yep.
I think it's an opportunity for us to say this is something holding us back from having this unifying set of ontologies, mechanism by which people are aware of the right ontologies to use for. You know, there's Uberon as one solution, but that's a sort of piecemeal, maybe underfunded solution. So we can identify the problem space, and then we can allow then the funders who maybe are looking at the reports to see where they see an opportunity.
I
Okay,
think
and
also,
certainly one could get support from various entities, right?
yeah, I think it might also be, certainly in my experience, a lot of the people who do the annotations are postdocs who are interested in a particular question, and they often don't actually see the urgent need to have a, you know, a common language, a frame of reference, because they tend to be interested in one particular problem. So I think there are also potentially issues around training, so that people really understand how important it is that there is this common language, and that is, if that's laid on top of an ontology, that can be very powerful for analysis.
I want to chime in with two comments. Two things we can pay more attention to. One is on the front line or upstage, where even before data collection, if we pay more attention on the metadata on, say, where in the microanatomy we cut the sample, or we make a good design decision to repeatedly go to, especially for animal models, to go to something others can repeat, so that there is a reference atlas, a spatial atlas that many people can chime in and can go back to do experiment again and again. That would help. Then on the back end, we are driven to make publication as an endpoint, but if there is supplementary support for this post-publication, you know, reconciliation of the taxonomy, that would be really helpful.
Yeah, yeah. I'm just looking at the time, and it's now 8, and I'm just wondering whether, so we've talked quite a lot now about basically very practical issues that arose in terms of cell type annotation, some of the roadblocks that we found. We've also identified some areas where things could move on. But I'd quite like to take the discussion now to the second point we had, and that was really, I'm just trying to find my notes. That was the other key issues that are likely to arise. For example, one of the points that was made earlier, that was mentioned right in the introduction, was how do we know what the ground truth is? How do we validate once we've come up with these cell type annotations? Do people have thoughts around that?
So one thing that we can potentially do is there are some cell types that are well defined. Everybody agrees. For example, endothelial cells, those are going to be present in every organ. Can we then look at the different technologies and see if they are identifying the same cell correctly? So we can come up with a list of cell types that could be analyzed by different platforms.
Yeah, but you also mentioned earlier, like the difference between a T effector and a T memory cell. So in some ways, it would be great to be able to actually have functional assays to link those back to, you know, just
sure.
Making those cell types.
Yeah, I think that's when, if you want to go down that far, yes, certainly you will have to look at the function. I was thinking just looking at just the highest level, like T cells, B cells, and endothelial cell, epithelial cell. Are we agreeing with different technologies? Are we seeing the same, characterizing the same cell?
Yeah, yeah. Actually, just from that point that you've made there very nicely, actually that leads, I think, very nicely also to the point in what type of reference data sets do we need to actually start to generate the cross-platform annotation? Do you really think it's something that we need to do exhaustively for every single tissue system? Or is that maybe also something where we can say, well, we'll try to do the cross-platform validation just on selected data and then try to infer for other tissue types?
Well, so the peripheral blood sample would be kind of easy to get and everybody can access it. And there are multiple different cell types there.
I agree, and that's great. The one problem with blood is that, at least with sort of, if you just take a sample, is that there's no spatial aspect to it. And I think a lot of the single cell studies are really moving from the dissociated tissue towards much more trying to understand each cell within the tissue context. And I suppose
Sure.
that's where blood breaks down a little bit. But otherwise, I agree completely. And I believe that some of the
another dimension of blood is if you think about just the vasculature itself. This came up in a different breakout meeting, but one option when thinking about ways to generate sort of ground truth or reference data at a single cell level across organs, it was brought up that perhaps thinking about vascular composition could be a component. So if you think about anything that's derived in situ that's histologically oriented on a slide or in some sort of volume, that spatial reference would be another potential thing to look. So even if not, you know, something like a blood draw that is in a liquid suspension, I think it's a good idea, as Anoop had said, to possibly consider maybe other constituents that are sort of scientifically cross-cutting throughout an organ system. And that could be a component of vasculature.
Yeah.
So actually we have a collaborative project undergoing right now in HubMap, where we're using the vasculature as the scaffold to map different cell types to. So we are going to be generating some data for vasculature segmentation. And we have in the past looked at for different markers, vascular cell maturity, how the cells mature over time. And all of this work we're doing by immunofluorescence, but the same can be done by other platforms, and then we can compare the results.
And when we think about the sort of cross-platform validation for a model system, what do people regard as a key problem that we could overcome by collaborating? Are there any ideas for that? Because it seems to me that it's very easy to fall back into your favorite tissue, and everybody will want to just go for that.
I think the key problem is that we can overcome is because different people have their own technologies and they are the experts. I cannot use somebody else's technology and come up with and compare it against mine because I am not the expert in that technology. So collaboration will help with that.
And I suppose that collaboration, we commented already on earlier that some form of finding out, good repositories of actual ongoing projects would generally be helpful. I just, earlier in the plenary, there seemed to be quite a bit of interest in incorporating this idea that we shouldn't just be annotating cell types on the basis of cell type, but that actually gene expression programs might be very, very important. And I think that also speaks to what Anoop was talking about, distinguishing different subsets of T cells. So they're all T cells, but within that label of T cells, they might have different gene programs activated to make them become a memory cell or an effector cell. And whether people have suggestions in how we could incorporate that kind of annotation into our cell type annotation. I think it's difficult at the moment, but I actually think it's a really quite an exciting way of thinking about annotation, because it would also, if we come up with something that allows plasticity, that would also really help us in terms of assigning cells that are transitioning from one cell state to another in terms of development. So going, sticking to the blood lineage, going from a double negative to a double more positive T cell and so forth. But that, we generate a system that allows us to label cells as they are transitioning. Okay, so it sounds as if this group does not feel that this is something we want to talk about extensively. I would, in that case, like to wind the conversation back to something that whether people do seem to feel more strongly about, and that was trying to match annotations between organisms.
And between modalities.
Yeah, and different modalities. So what do you see as a key issue? Let's start with the organisms. So what do people see as key issues when we try to annotate between organisms?
So I think part of the problem is that we're really getting into the deep biology. At least the efforts to go between human and mouse with the developing kidney. It was, I think you have to keep a fairly high level of organization to this, or at least not try to get too far down into the details, because I mean, it was amazing that in the developing kidney, of the 23 transcription factors, which are thought to be really key in identifying the various kidney cell types, only half of them are actually present in the human kidney, a developing human kidney, and several of them are being expressed in actually different cells than what would have been predicted from the mouse. So again, there's going to be a lot of devil in the details of people who have expertise both in the human side and in the mouse side.
Just to follow up on that, even though the transcription factors were different, did you still recognize the same expression modules that were activated in these developing kidney cells?
Yes, and what was amazing was that in mouse, if you think about, say, 8 steps in the formation of the nephron, where you're getting self-movement and changes in gene expression, and you can identify various segments, maybe that's the best way of putting it, over a time course. When you then look at human, as it turns out, the developmental process is extended. And so what looked to be going from, segment 3 to segment 4 in mouse was actually segment 3, 3A, B, C, 4. So you actually have more details in the changes in the morphogenic changes that go on when you look at human versus mouse. And then that helped to inform going back to the mouse studies to try to look more closely to see, can you actually capture these additional steps?
And I suppose you bring up development there, and I think that in terms of cell type annotation, there's another whole set of metadata that actually at the moment, certainly in the cell type annotation, we are not capturing very well, and that's the time component. How long does it actually take a cell to transition from one state to the next? How coordinated is it throughout development? And also how much variation is there? Is that a very consistent program?
So, I mean, from at least from my perspective, when I talk development, I try to tell my folks that development is just not embryogenesis. It's the entire life history of the organ, from cradle to grave. And so even within the context of HubMap and certainly within HCA, being able to be able to parse out, this was from a juvenile organ, middle-aged organ, an old organ, of the same organ, because I still think that there is the aging process, which I consider, kind of part of the development process, where you do expect to see program changes reflective of the aging of the organ. So I'll stop there. I'll get off my soapbox.
No, I think it's a very important point, and I don't think we're capturing that very well yet. So maybe we can record that as a problem that we don't really have an answer for yet. And I guess it will be, it is also not easy by just saying, oh, we'll just collaborate more and we'll find it. I think we'll need to generate a lot more data before we start to get a handle on that kind of spatiotemporal aspect of cell type differentiation. Do people have particular favorite techniques that they would like to integrate with single cell data but are finding it difficult? Are there particular groups here present and represented that feel that the way the discussion has been going at the moment and talking about cell type annotation, that maybe some aspects that they're encountering has not been highlighted? I mean, I very much come from the single cell RNA-seq world. Is there anybody here who might like to comment on proteomics, for example?
Oh, hey, can you hear me, Kerstin?
Yes.
You said the magic word. I'm a labeled proteomics person, and I'm interested in like how functional clusters of cells will bubble out of this process. And single cell proteomics is on the lips of many, but it'll be some years, I think, before we can get the density of data that single cell RNA-seq can. The numbers of cells and the depth is, of course, giant technological challenges for single cell proteomics. So in the interim, having functional clusters that have identified where you can get, say, 100 to 1,000 cells, and through laser capture microdissection or other approaches, start to probe the biology in, I'm going to be a really avid consumer of what comes out of cell annotation and cell types. I also think that we need to determine all the proteoforms that are expressed in each cell type. So to regularize the human proteome in that way. So I'm eagerly awaiting how the field sort of lands on these annotations. And, so like islet cells in the pancreas, right? Everybody gets that. And how will this group expose functional clusters? Because those are actionable, at the 1,000 cell level, even in the very near term. So just two general points. I can ramble on as long as people wanted to listen.
No, I think they were very, very good points. So first, that of course the technology maybe hasn't quite reached the same throughput, but that of course we absolutely, at the moment we pretend a cell is a sort of bunch of RNA, but of course it isn't. It's the proteins who do the job, and really we have very little handle on, different protein isoforms or post-translational modifications, and there's obviously so many more layers that will eventually be considered, but that are probably just a little bit beyond our technology at the moment. But maybe if we can come up with good ways of annotating the cells for the technology that we have at the moment, then maybe those can in the longer term be rolled out to other modalities as well. And I have no doubt that as we start to have those additional layers, we'll be able to get to the more fine-grained level. And to me personally, it comes back to those hierarchies being very important, that initially we might just cluster on more coarse grain, and then as additional layers of technology are being added, we can in a way subcluster that. And I also think that those subclusters will then reflect certain gene programs and responses to certain stimuli. What are there any, we haven't really talked much about any spatial data, but I guess the cell type annotation there, by and large at the moment, concentrates on, again, finding the right cell types. Are there efforts being made to understand what proximity, measures of proximity of particular cell types next to others, or that kind of data? If somebody could comment on that.
So, Kerstin, this is Anup. So we use immunofluorescence, multiplex immunofluorescence. And we've been doing some neighborhood analysis just to see what cell types are located next to our cell of interest and how that influences patient outcome. Most of our work is in an oncology area. So we're essentially looking for, does that, how they, for example, clustering of immune cells next to the epithelial tumor cell, does that predict a better outcome or not? So we are trying to ask those questions. Other things we are looking at is identifying regions of hypoxia and seeing how those, not only the size of these regions, but number of these regions within a core or the tissue section, how that relates to outcome. So those are the things we're trying to address right now. And obviously there's a lot of work going on looking at just the heterogeneity, the Shannon index, and correlation of that to outcome.
Yeah.
But I am very interested in figuring out how to relate the cell types that are determined by RNA-seq to the cell types that we detect by immunofluorescence, because we are not able to do thousands of proteins in situ. And the cell types that are being defined by RNA-seq generally takes many transcripts to define a cell type. And the RNA does not always translate into proteins. So we are struggling with some of those issues.
Yeah, no, that makes a lot of sense. And obviously the proteins are also much longer lived, and you might only have very small changes in RNA levels, but very profound changes in protein abundance. I mean, everybody who clusters T cells knows that CD4 and CD8 T cells don't always separate terribly well, and yet at the protein level, they're, you know, functionally incredibly different and well defined as cell types. I suppose one of the things that is really going to help with cell type annotations there is an increase in techniques that really do both, a little bit like CiteSeq, where we are now starting to look at up to 200 antibodies or so that can then define the expression of 200 different proteins in the context of the transcriptome of the cell.
So is that being done on the same cell, transcriptome? Yes. And oh, that's great. Good.
Yeah. And I think from what I hear, that is, it takes a bit of optimizing, but there are now commercial panels available, and that seems to be working quite well, at least in some labs.
What is it called? CiteSeq?
Yeah, C-I-T-E, seq.
Okay, thank you.
Right, so we have about 10 minutes left before the session is going to break up. And I just wondered whether we could help the people who are trying to wrap up the whole meeting to see, have a look at what we've actually recorded as our outcomes.
So,
Kathy, have you?
Yeah. Yes, if I could actually just walk people through, maybe I should share my screen, and several people have been helping with the note taking, so thank you very much. But I'll walk through the topics that I identified, and then we can decide which ones should end up in the blue boxes.
That would be really helpful. Thank you.
Let me just make sure I'm showing you the right one. So are you seeing right now problems we think we can solve by collaborating?
Yeah.
Okay, great. So the first topic I identified was identifying ontologies that we can use cross-species and cross-modality. Is that something we want to list as a follow-up action to this? And a lot of this is notes about the discussion, but the questions that we need to answer are, when will we meet? Who's going to lead this group, this discussion? And part of this discussion was about doing sort of a minimal, talking about the possibility of an upper level ontology that people would use to capture the terms that would allow us to go across species and cross modality. Sorry.
To me, it almost came across as if one of the key problems was that, in a way, a step back from that we all know ontologies exist, but actually its use is really not widespread enough in our community, and therefore also not yet being applied to the cross-organ, cross-species and cross-modalities, because people are still very much approaching things from their own biological question rather than trying to put things into that wider context.
Okay, so does that mean that there's no value in trying to come up with a common nomenclature that some
No, 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 absolutely not. But that I was agreeing with you that actually, yes, I do think this is a real problem, but that we definitely need a common nomenclature, and also that there's still some work to be done to educate people to try to use that as much as possible. And I think I'm guilty of it too, that when we annotate cell types, we just say, oh yeah, that's a B cell. And one day we call it a B cell, and the next day we call it a B lymphocyte or whatever it is. So that yes, the field needs to make a more concerted effort for controlled vocabulary.
So is this a problem that we think we can solve by collaborating?
Yes, I think by bring, oh, there's somebody in the chat. Is that, sorry, this has just been blinking at me. Oh, okay, no, that's slightly site discussions.
I can't see everyone, so I can't see.
So yes, I think this could be, so maybe one of the things we could list as, you know, a problem is defining and using a more controlled vocabulary.
And are you seeing this blue? I just want to make sure you're seeing the blue box now that I created for this.
Not on the screen, but I can.
Hold on. Let me. Apologies, everyone. How about here?
Ah. Perfect. Yes, I think that sums it up very nicely, actually.
Okay. Is there anyone? Are we looking to add names to this at this point and propose when there would be a next meeting and what additional expertise is needed? Is this what we should do at this point?
I guess it is. So because this is really cross consortia, I think I would say, you know, reach out to future to leaders of consortia and conference organizers to, you know, reiterate this point and
consortia and sorry?
Lead out to readers of consortia to lobby funders, I would say, because, you know, one of the point we're making a few sentences further up is to say that, you know, having funding to support these efforts would be really helpful.
Okay.
In previous breakout sessions, I was present. People were also asked at the bottom of the document to write down their name. So if people don't do this, obviously on the screen that Kathy is showing us at the moment, but if we go to the actual document, and the link to this is in the chat. So
yes, and actually it might be useful if people wanted to, oh, sorry, names with consortia on the team. So this sounds like it's assuming that the people, I think that we need to have a list of people who are interested in joining this subgroup. And I'd like to suggest that the list go here. And people can just add their own names?
Yes, that would be great. I will add my name when maybe after the meeting, but so I think it would be really great if everybody could just take these last few moments to you know, express an interest in this. Does everybody have the link to the document? Just in case they don't, I will just copy it again from the site and put it into the chat.
And there were a couple of others that I captured there. So once you do that, I thought I would just mention the others that I captured, and we can correct them if these are not correct.
Okay,
right?
Yeah, that would be great.
So another one I collected was improving the ability to share tissue so that we can collect data from different modalities for the same tissue.
Yep.
Is this something that this group feels they can work on? what additional expertise do we need? We need representatives from different organs, is that correct? Or from different groups that are involved with the ability to reuse the tissue?
So I would be happy to put my name, but this is,
I'll put you in.
yeah, so.
Are you referring to disease tissue or normal tissue?
It doesn't matter either. So I was going to put at the back here, so so I think what I could contribute here is working on building a platform within the HCA website, for example, that is widely available. You don't need to be a member of the website to share this information. Yeah.
Ellen, I'll capture that information. I will go through the chat and capture any information that I've missed. But if people could go on to this list and add their names where they'd like to contribute. I'm going to go back to, let's see, are those, oh, those are the only two that I have listed. So let's go back up to this list. Do we want to have an action around reference data sets?
I have to say, when from the discussion, there wasn't a particular action item that was obvious to me, because I think, yes, blood is a useful reference set, but blood is already being widely used, I guess. And I got the impression that this is probably, again, something that is going to happen within biological networks for specific tissues. But please, others chime in if you feel differently. Does somebody know when we're meant to be going back to the main plenary?
It was just a note that we have less than 5 minutes.
Ah, okay.
What about these other topics? What about collaboration helping with tissue-specific technology? I'm not sure I captured what that discussion was getting back to. Is there any action there? Item 5, was there any action around this? Should we capture cell type annotation, different gene programs?
No, I don't think we decided on any action on that.
And then matching annotations across organisms and modalities. I feel like that gets back to the ontology question. Was there some other subtlety here in action that we should identify?
I think it's useful, actually, even if we don't, I mean, obviously it's ideal if we find a action item that we can take out of this, but having some of these issues raised as problems that the field is discussing, I think that's valuable in itself. I think we also have to be slightly mindful of this fact that we've now all spent 2 consecutive days in video conferencing, and I fear there's the contribution from the different members in this Zoom room is probably, you know, people are flagging a little bit, I fear.
Okay.
I can help with tumor tissue.
Thank you.
So should I capture that in one of the actions, or?
Oh, I can send you, I send you a text.
Okay.
I work at a cancer center, so we have tumor tissue to provide with MTA.
Thank you, that's great. The next one, proteomics, is relatively new, but interested in what we're doing. I don't know that there's any actions there, except maybe we want to reach out to include proteomic people more often, or maybe it's too early.
I think really it's useful to be aware of all these technologies that are coming up, but in terms of cell type annotations, I think the issues that we talked about, the different layers and hierarchies of cell type annotations apply just the same. We didn't really get onto how do we validate these, because that's another whole field of, you know, functional validation. So I don't think we need to capture a specific, you know, action for everything we talked about.
Okay. And then the spatial information, is there any subgroup that should follow up on spatial information?
I believe there was a session on spatial analysis, spatial profiling yesterday, so maybe they have captured this.
Yes, so we should liaise with them, because I feel this is actually incredibly important, how we start to think about, you know, mapping cells in space.
Great. I think we're going to be pulled back in a second. So I just wanted to say that I will go through and clean up these notes a little bit. And Kerstin, I'll work with you on that to see if I've got everything I'm sure I missed. And for those of you who've been here, please don't hesitate to go in and correct and modify my notes. Thank you so much.
And thank you so much for volunteering and taking fantastic notes. So that's great.